Alright, this is Jahangir from MedPorium, and today we're back with part 4 of the urinary system. In part 4, we'll discuss the third and final step of urine formation, which is water conservation. So if we're going to talk about water conservation, we need to talk about the part of the nephron that performs this action, which is the collecting duct. Now at this point, we talked about the collecting duct many times throughout the series, and you should know the location of the collecting duct and its functions. But let's just use some active recall to recall this knowledge. The collecting duct begins in the cortex of the kidney and receives tubular fluid from many other nephrons at the same time. They concentrate the waste to become urine and control water loss by reabsorbing as much water as they can, depending on how hydrated the person is. The collecting duct does this and produces a hypertonic urine, meaning there's a high osmolarity in the urine. To further talk about the collecting duct, let's move over to the Thinkpad. Alright, so we're at the Thinkpad and we want to understand the functions of the collecting duct, just to review. So there are two things that allow the collecting duct to produce hypertonic urine. The osmolarity of the lower medulla created by the nephron loop is part of that. So as we talked about the functions of the nephron loop, we said that it creates an osmotic gradient. This osmotic gradient influences the reabsorption of water, which in fact produces hypertonic urine. The second thing that allows the collecting duct to produce hypertonic urine is the medullary portion of the collecting duct. It is more permeable to water, meaning this part of the collecting duct is more permeable to water, letting water go in. This allows easier passage for water, which influences water reabsorption and overall produces hypertonic urine. Alright, so we just wrapped up our conversation about the collecting duct from the Thinkpad. Now let's talk about control of water loss, and to do that, we need to move over to the Thinkpad. Alright, so we're at the Thinkpad and we want to understand the control of water loss. Well, to do that, we need to understand that concentration depends on hydration, meaning the concentration of your urine depends on how hydrated you are. So let's just say you drink a lot of water, which means you're very hydrated. You produce a hypotonic urine, which is also called water diuresis. Now when you pee, your urine will be more in the lighter yellow. Now this stimulates the collecting duct to reabsorb more salt to maintain the medullary osmolarity. Now normally the collecting duct reabsorbs water, but in this case you're very hydrated, so it wants to reabsorb salt so it can maintain that medullary osmolarity. The whole objective is to maintain the medullary osmolarity. Now this is opposite in dehydration, in which urine is more concentrated than usual, which with a high osmolarity, which is hypertonic urine. In this state, the pituitary gland releases antidiuretic hormone. Now, as we talked about in part 3, antidiuretic hormone increases water reabsorption by stimulating the cells of the collecting duct to transfer aquaporins, which are channels of water, from the vesicle of the cell to the apical cell surface, which reabsorbs more water from the tubular fluid. So the apical cell surface is just the cell surface closer to the tubular fluid. Now second, if antidiuretic hormone levels remain elevated for over 24 hours, the cells produce more aquaporins, raising the water permeability of the collecting duct. So just remember, concentration depends on hydration. That's the one thing I want you to learn and take account. Alright, so we just wrapped up our conversation about the control of water loss from the Thinkpad. Now let's talk about the Countercurrent Multiplier. Well, the ability of the collecting duct to concentrate urine depends on the osmotic gradient of the renal medulla, as we talked about before, which is controlled by the Countercurrent Multiplier. So what is the Countercurrent Multiplier? Well, it is a mechanism that recaptures salts and returns it to the renal medulla, creating the iconic high osmolarity. So if you want to review or just try to memorize the countercurrent multiplier, think about the recycling logo. It's kind of recycling or recapturing salts to create the high osmolarity of the renal medulla, which in turn helps reabsorb water by the collecting duct. To understand the countercurrent multiplier, let's move over to the Thinkpad. Alright, so we're at the Thinkpad and we want to understand the countercurrent multiplier. Well, to do that, we have a picture right here, and we'll start by number 1. So, more salt is continually added to the proximal convolute tubule. So, let's just say this is our salt, just going through and going through. Now, the high osmolarity of the extracellular fluid, the more water leaves the descending limb by osmosis, meaning the high osmolarity out here, the more water leaves. Now, the more water that leaves the descending limb, the saltier the fluid is in that remaining tubule, meaning as water leaves, the tubular fluid becomes very salty, as shown by the 1200 osmolarity. Now it gets saltier and saltier in the ascending limb, and the more salt in the tubule pumps out in the extracellular fluid, meaning that in the ascending limb, more solutes are pumped out instead of water. The ascending limb is impermeable to water, but permeable to solutes, as I talked about before in the series. But the descending limb is more impermeable to solutes and permeable to water. Now this is kind of like a balance, like a seesaw. One goes up and the other goes up. So more salts are taken out, creating that iconic medullary osmolarity, and the more salt that is pumped out of the ascending limb, the saltier the extracellular fluid becomes, and the renal medulla becomes more salty, creating that osmolarity that we all know. Now, as we go up to the distal convolute tubule, the tubular fluid becomes more diluted, since the ascending limb is more permeable to solutes, letting the solutes go out, but impermeable to water, leaving water going up to the distal convolute tubule. Alright, so we just wrapped up our conversation about the countercurrent multiplier from the Thinkpad. Now let's talk about the countercurrent exchange system. Now, the countercurrent multiplier, countercurrent exchange system are basically the same, but the countercurrent exchange system involves the vasorecta and its effects on maintaining the osmolarity of the renal medulla. To talk about the countercurrent exchange system, let's move over to the Thinkpad. Alright, so we're at the Thinkpad and we want to understand the countercurrent exchange system. Well, I have a picture on my left and a description on my right, so let's talk about the description. So the vasorecta carries large amounts of water, reabsorbed by the collecting duct, and forms the countercurrent exchange system to prevent themselves from also carrying salts and urea. Now you might be wondering, why is the vasorecta even here? Well, in our anatomy portion of the urinary system, we talked about the vasorecta being the blood supply for the medulla, so it's inevitably going to interject. Now, the countercurrent exchange system prevents them from also carrying salts and urea, which maintains the osmolarity. If they did take salts and urea, we wouldn't have that osmolarity and we wouldn't have water reabsorbed, which would dehydrate us, and our system and our body, our tissues wouldn't get the necessary water we need. So at this point, you might be wondering, why is urea being reabsorbed by the collecting duct? Well, urea is permeable in the collecting duct, allowing urea to go through. But don't worry, not all urea escapes. Most of it stays in the collecting duct, in the tubular fluid, and goes out as urine. Now, urea, once it is reabsorbed, it usually goes up through the nephron loop, since the nephron loop secretes urea, and then goes all the way around anytime it wants to. The benefit of having urea reabsorbed is to create that high iconic osmolarity of the renal medulla. So that, ladies and gentlemen, wraps up part 4 of the urinary system. In part 4, we discussed the third and final stage of urine formation, which is water conservation. As always, a pomodoro a day will make you a doctor someday. I'll see you next time.